also to climb and to eye optics. I'm really happy to be here. But before I start mumbling about my work, I would like to know how many of you know deconvolution? Yeah? Good, quite some. How many of you have worked with microscopy data? Either created or seen or analyzed? Okay. How many of you have background in computations? Okay, so there are quite some good points to connect. Good. I am going to talk about some fancy words here, which we will learn soon. So don't worry about the long title. Let's go into something more interesting. We are here today somewhere, and it's one of the top universities of the world, right? But I want to challenge you. I come from the topmost university of the world, at least geographically. We are working on the rankings, but geographically we are the topmost university. And what I'm trying to do here is I'm trying to show some beautiful pictures of Tromsø to lure you in. We really like to work with best talent, so come for the Northern Lights, stay for research. There I work with 3D nanoscopy. This is a group that I founded, and as it says, we do not microscopy but nanoscopy, and we like to work in 3D for living cells or living biological systems. A quick introduction about the range of activities that we do. Our primary research funding is about new label-free imaging technologies. And we make new instruments, but also new algorithms. The core idea behind this activity is symbiotic development of the equipment, the algorithms, and the application. They should feed.
I'm sorry, did you need one of these, the clicker things?
I got
Okay.
it from.
All right,
Thank you very much. Yeah. So basically, we want the instruments to be designed for algorithms, and vice versa, and they should have an application-sensitive development. But we also work on fluorescence computational microscopy. This is where I started at UIT. And this area has flourished a lot, and we will see most of the results in this area today. But I'm also very passionate about building the bridge between AI, microscopy, and life sciences, mainly because life scientists have been using microscopy for a long time, but they need to get information and knowledge out of it. So I have learned that I can create value there by actually bringing all of them to the same platform and bridging the knowledge gap, the language gap, and so on. So this is something that we also work extensively. And as a result of this, my team, 3D nanoscopy, is extremely multidisciplinary. You will find a biomechanic in our lab, you will find a pure chemist in our lab, you will of course find computational scientists in our lab, but also people of different backgrounds. We really like mentoring young researchers. Our Ph.D. students and postdocs all have career development plans that they have developed on their own, based on their vision of their career, and we help them in realizing them. And we work on innovation, so we have some patents and pre-commercialization activities. But my core strength is in modeling light-matter interactions, and this is what I will talk about in some depth today. Let's say that we have a region that we want to image. It's called, it can be called a scattering medium if there are things that can scatter light. Now, if there are only a few scatterers in this, then the input light can be modeled with something called transfer of intensity equation, or with conventional EM theory. So if some of you have been working in research with computational microscopy, you would have heard of Born approximation, or linear models of light, and these fall here. These have been developed in the microscopy community for a long time, and I am not needed for this part. I am rather needed for this part. Full wave multiple scattering EM theory. What happens is that if we have a medium with many scatterers, the light usually doesn't go ballistic or doesn't follow simple paths. The light follows a pretty complicated path where the light bounces off multiple scatterers, creates near field interactions, and then the information is seemingly lost. The funny thing is it's not lost, it's encoded differently, and this is where my expertise of working with EM theory comes into picture. Another place where I hope I can create value in the microscopy community is for extremely highly scattering medium, where neither the simplified transfer of intensity equation nor this full wave multiple scattering EM theory works, but we need to go for the full-fledged radiative transfer equation. This is a very new topic in optics domain, and it will take many years to come through, but we have started foundational work on this. And of course, the staple fluorescence microscopy is something that we work on. But what is important to understand is that as a consequence, we work on any kind of incoherent light phenomena. A little bit of the layout of presentation, simply to put some words in your mind to look forward to, but I have not necessarily segmented my presentation along these topics, they flow into one another. So we will very briefly discuss how I think of computational microscopy, and then discuss an algorithm musical that I invented in 2016, and it has flourished into many different directions, including super resolution, motion resolution, and example imaging. And then we will work on the new groundbreaking work that is happening in my group on coherent label-free microscopy. So what is computational microscopy? A computer and a microscope. One may think so, but I have put a question mark here because I think it's a three-piece puzzle. Do you know what this puzzle, when integrated, will look like? We need to be able to use these three pieces to create this shape. Computational microscopy is very much like this. We already know that there has to be a microscope and there is a computation. But the thing that binds them and makes this very interesting 3D structure is physics. In computational microscopy, physics is the most important element that integrates all of them and separates it from image analysis, for example. With this, I am moving, the reason, yes. And although you may think that this computational microscopy is a very fancy topic, very difficult topic, in fact you have used it. If you have used deconvolution, you have worked with computational microscopy. This is the original image of microscopy data, and this is the computed image. But the thing that brings physics in place is this 3D point spread function. So when you computed this deconvolved image, this point spread function of the microscope, which represents the physics of your instrument, is used in the background for computation. So you have been using computational microscopy. Have you heard about single molecule localization microscopy? Yeah, I see some nods. And there also we use physics in a very unconventional sense. Let me first take the microscopy and computation part out. This is a microscopy image of a single fluorescent molecule. If you have a microscopy image of a single fluorescent molecule, you can do something called center finding using Gaussian fit or another kind of fit. And after finding the center, you can say that, yeah, this is where probably the molecule was with certain precision, right? But this is the case if you have a single emitter. If you have many of them labeling a structure, then this property cannot be applied anymore. You see a blurred structure of combined emissions from all the molecules that are present. Now, we introduce a physical manipulation by actually changing the chemical labels. Instead of using regular dyes, we use their special dyes, which are not emitting light most of the time, but sporadically, stochastically emitting light in certain bursts. This means that we can physically time multiplex the information. A lot of molecules remain dark, maybe just 2 or 3 come up. Now we have a case of an image of a single emitter, and we can perform localization. At another time instance, other few molecules light up, and then we can repeat it. So this is the concept behind single molecule localization microscopy, where the binding physics is that we can time multiplex the photokinetic behavior of the molecules. But what I have been interested about is, can we use the physics of the sample itself? Instead of changing the fluorescent molecules, can I use some property of the fluorescent molecules themselves? And in that quest, I made this algorithm called musical. And in order to explain this algorithm, first let me explain the physics of these molecules, this sample that I'm talking about. Consider a petri dish which has cells, and then in cells we are looking at certain structures, and we have labeled these structures with fluorescent molecules, right? We put this dish into the microscope, and then we get an image. But instead of taking one image, if I were taking a video of it, I would see a very interesting phenomenon. Almost all the images look similar, but they are slightly different. Can you imagine why there is a difference? Noise is one of them, right? But there is another reason that they are different. Each molecule does not give exactly the same number of photons out every single time. This means that each molecule has a slightly different temporal behavior in every frame. This is an equivalent example. We have a bowl of rice, and we have a spoon to scoop out rice, right? The size of the spoon is constant all the time. This is equivalent to putting in the same number of photons onto the sample all the time. But every time, if I were to scoop 1 spoon of rice and count the number of grains, sometimes maybe there are 50 grains, sometimes maybe there are 48, sometimes maybe there are 56, and so on. So there is going to be a fluctuation. But what is interesting is that there is a fluctuation which is systematically encoded via the microscope. So we have 2 types of fluctuations naturally present in a video data. Fluctuations because of the nature of fluorescent molecules, which get systematically encoded, and fluctuations due to noise, which are totally stochastic. This is the property that musical uses. What musical does is that it takes this video stack and puts it through an eigenanalyzer. Eigenanalyzer is akin to principal component analysis and other such techniques that you may be aware of. What eigenanalyzer does is it makes something called eigenimages. I will show you examples of eigenimages later here. And each eigenimage is assigned an eigenvalue. If there is a high eigenvalue, it represents a statistical feature which is consistent through the data, through the video. What would be consistent? Systematic encoding, right? So the things that gets encoded systematically are collected into a set called phi signal. They all have high eigenvalues. But then the non-systematic ones have very low eigenvalues, typically close to 0. So we can pull them into another set called phi noise. This is a systematic pattern that can appear from an image like this or a video like this. This is another one, a third one, a fourth one, a fifth one. So all of these are eigenimages of a video such as this. On the other hand, the noise eigenimages just look like noise, right? So this is the first operation that we do in musical. We separate systematical features from the completely random features. And then we do something called computational confocal scan. We take the point spread function of the microscope and we scan it or we convolve it over the eigenimages, all the eigenimages. Now when we do this, we get two new images. The first image is a confocal scan on the systematic patterns, which then gives something which is similar to denoised deconvolved image, because all the noisy patterns have been taken out. So it's just doing a very fine confocal scan. But this is more interesting. When we do this computational confocal scan over noisy patterns, then it gives some random values everywhere, but it gives 0 at the places where there was a systematic contribution. And this 0 is like a very sharp 0, it's not a blurred 0. So from these images, this is sharper, but not as sharp as this. It's a negative image, but a sharp image. So this means that here we have deconvolution, but here we have anti-super-resolution. And we use this anti-super-resolution by taking a division. So we divide this deconvolved image by this anti-super-resolution image to get very nice super-resolved image where there are peaks at the location of systematic source. Yes?
But is that because where those objects are, it's a different noise pattern? So there's still noise there.
There's still
but
noise
it's just
there.
at a different spatial frequency, or
Yes.
What's different about that makes it stand out?
Yes, so basically let's say that we had a molecule here, and this molecule had fluctuations, right? And those fluctuations cause these systematic patterns, but then there is also noise on top of it, right? So this noise will also be present at the location of the fluorophores, right? But what has happened after this separating of this is that the systematic portion has already come out, and then the non-systematic has remained, right? Now when this non-systematic has remained, this non-systematic one has a different contrast than the non-systematic ones around. And this different contrast is what works with this confocal scan to say it's 0. It's a mathematical property which says that anything systematic will be absent in these. So
isn't it still like the centroid finding, like the Gaussian finding, where you're trying to localize where?
It's almost centroid finding, but it's so in centroid finding we are really looking at the center, right? But in this case, when we do this kind of confocal scan, it automatically, mathematically converges to the place where there is a 0, where there was a systematic pattern. Yeah, but the concepts are similar. So we can take an SMLM dataset, single molecule dataset, repeat this, and exactly the same thing will happen, that instead of centroids, we will have zeros at the location of emitters. Yeah.
But is that only still, maybe we'll get to this, it only works for sparse?
No, it can work
so
on
it's
extremely dense samples.
Okay.
That is the strength of it. Because in musical, it's looking for marginal fluctuations. Even if there are marginal fluctuations of the order of noise, but they are systematically encoded, they will be separated. So we have been able to work with extremely high density samples and signal to background ratio of 2. So it's like any kind of blinking, etc. would be actually washed out. I will show you an example. Yeah. So this is the example. This is a mitochondria. Yes.
Sorry, I had one question.
Yes.
In the eigenal analyzer where you separated the eigenimages out, how do you actually decide which ones are the signal images and which ones
Yeah.
are the noise ones?
A very good question. So we look at their eigenvalues.
Okay.
Okay, so I do not have a plot here, but the eigenvalues clearly fall into two clusters. So we cluster them and then we see that, okay, these are high eigenvalues, they fall in one, and then the low eigenvalues fall in the other, and then we threshold that. That was the first thing we did. But then we made our algorithm parameter free, in the sense that instead of saying that we have Poisson noise here, so basically the noise characteristic actually depends on also what number of emissions were there, right? So instead of saying that we have a very neat segregation of the signal and the noise, we have now resorted to something called soft threshold, which says that, you know, they may have some contribution in signal through, you know, the extra fluctuations coming from Poisson noise. So we then, instead of separating it like this, we say that, okay, we perform deconvolution and we give these images smaller contribution here and these images smaller contribution here. So we have made it parameter free as well, so that it's a smooth transition between these two spaces, to put it that way. This is a mitochondrion. Inner membrane was labeled. And you see that it's a very, very noisy image. The signal to background ratio was very low. The reason is that this is 2 milliseconds exposure. If we compute its diffraction limited image by taking average over many, many frames, then we get an image like this. But if we perform musical on it, this is what we get. We were not sure if we are actually looking at something called cristae, but the biologist was sure that these are cristae, but the biologist was not sure whether there are some meshing artifacts here. But these meshing artifacts are like almost removed when we use this soft thresholding that I was telling you about.
This was fluorescently labeled.
This was fluorescently labeled. It
So
was,
are the bright spots corresponding to the fluorescent molecule?
yeah, so everything that is bright here is fluorescent molecules, but these extra bright spots are probably the locations where there were more fluctuations. Musical is a fluctuation sensitive algorithm rather than quantitative or density sensitive algorithm.
I see.
Yeah. And that is the yeah.
Can I ask a question? Have you compared this with the sparse localization and
Yeah.
see if the structure matches?
Yeah. So we have done extensive tests on single molecule data sets. And in single molecule data sets, of course, the precision of each single molecule is 20 nanometer or so when we use SMLM algorithms, right? But what musical is good at is taking structure out of them, statistical consistent pattern out of them. So a microtubule in SMLM dataset looks like spotty version, whereas musical result of it looks like straight clean microtubule. So that is another thing where musical is slightly different from SMLM. And some people like structure, but some people like specific molecules. So, you know, it's a matter of choice. Now I said that musical works with fluctuations. And the first thing that I presented is the natural fluctuations in the number of photons of fluorescent molecule emits. But musical can work with a variety of fluctuations. It can work with motion, it can work with noise. Noise is also a fluctuation. So something being stationary is not fluctuation, but noise is fluctuation, and that can be separated. It can also work with fluctuations created by illuminations, systematic or non-systematic. And I am going to present some of these results after discussing with client members and seeing what they have done. So this is mesoscopic imaging. We have probably not mesoscopic scale, but we were able to image half a millimeter by half a millimeter slice of fish cells. And these are living fish cells. I should have gotten video of them, so they just flood like they are like marathoners left loose. But basically we had many cells, and we used chip-based nanoscopy, where there are multimodal illuminations which keep fluctuating. So it's like illumination that is fluctuating, and then we perform musical on them. So if we take the diffraction limited image and perform musical, we of course get a much better image. But then we can zoom in and perform 60x image, for example. The features look comparable, except that musical says it has less background. And then we can of course perform musical again to have much sharper and super resolved image, for example, these regions. This is one. So for example, if there is a mesoscopic system and there is a way to introduce illumination fluctuations, methods like musical can be directly adapted. This is another example where we had a muscle tissue. Muscle tissues are fibrous tissues, and then if we take a cross section, then we see these very nice structures. And this is a cross section, so we have taken a cross section like this, and we have put it on chip-based microscope and imaged it. So you will see these structures. These are the ones that we see here as well. Now it is like unfair to compare this region because the contrast is evident. So I will rather compare this region where the contrast and diffraction limited and musical seems similar. Yeah. So the top one is diffraction limited and the bottom one is musical. And we see that certain structures are pretty well resolved. At present, we do not know how to use them, but one of our university spin-offs is considering how to use this information for pathology. Another source of fluctuation is motion. What if there is a point-like object which moves from one place to another? If you look at the microscopy data, a video equivalence would be, it's like capturing this vesicle or this point-like object in different frames. So at some point, the molecule is here, in another frame the molecule is here, and so on. So it's like evidence of fluctuation. It's a spatial fluctuation over time. And if we apply musical on such a data, musical cuts out the structural information and instead retains the motion trace. The reason is motion is a bigger source of fluctuation than the structure itself. And this is a result that you had seen in yesterday's presentation, at least some of you, where we had this vesicle which was going from here to here. And then when we perform musical on it, we see this trace of its motion, and we also see its hopping positions. So the vesicle itself is pretty large, but the motion of vesicle is traced with super resolution. We were able to also apply it to label free. So we did QPI of moving beads which were optically trapped on a waveguide and propelled through the waveguide length. So this is then again a point-like object moving across this direction in label-free imaging mode. And then after applying musical, we could get its motion trace, including the positions where there was a hopping or a fluctuation in its position. It's like trying to hit the boundary and finding a different route. We have also started working on thick sample imaging using musical. So this is a kidney organoid which is mimicking the kidney tissue environment. And the big structures that you are looking at are glomerulus, but the interest is to look at the small structures, podocytes. This is 3D sim. We took exactly the same 3D sim data, no separate measurements, but instead of using their proprietary algorithm, we applied musical in 2 different versions. And as compared to 3D sim, which has so many artifacts, because it being large, because the illumination being variable, and because that the signal to background ratio was poor, we have a lot of artifacts, but in musical we can remove those artifacts significantly and look at podocytes pretty cleanly. But moving on from musical to other things. So when we have a thick sample, even if we use the most optimized microscope, one of the problems is that the sample itself creates scattering and aberrations and unknown, unqualifiable forms of optical manipulation. So instead of a spherical object looking like this or like this, we see non-uniform aberrations of different kinds. And due to this, the images of thick samples are typically much more blurred out than equivalent thin samples. So what we have been working on is to reconstruct the point spread function from the images of the samples themselves in order to improve their image quality, which means that we do not characterize the microscope, we do not compute its PSF. Even for doing deconvolution, we learn the point spread function from the image itself. So this is a 20 micrometer thick sample. It's organoid at a smaller scale. It's a public data, so we have no influence or no knowledge about what parameters were used and whether there was an optimization. And this is the kind of blurred image that they have. But after applying another state-of-the-art algorithm, which was proposed by the people who shared this public data, we do get much better contrast. But do you see these artifacts? Do you also see these out of focus regions coming into the image? These are removed when we use our point spread function reconstruction together with deconvolution. I'm not discussing too many details about it because we applied for a patent for this one.
So that's still acquiring data where the light and the beam is incident from above. So
Yeah.
that's just not differences in attenuation between the two systems, because those are different imaging systems. I don't know,
Yeah.
different wavelengths.
Definitely. So if we change the imaging system, for example, instead of going for this epifluorescence kind of illumination, if we were using confocal, or if we were using multiphoton or whatever, in that case we have at least solved the illumination created optical blur or illumination created variation, but still the emitted photons need to cross the sample and come out. So there is still some blur, right? And that is PSF characterized, but then in this case the PSF is determined on its local sample environment, and that can be learned and compensated. So in this case, it does not matter what microscope you have. It does not matter whether it's label free or labeled or coherent or incoherent. If we know some basic point spread function model that is relevant, we can learn everything else and then we can compensate for it. And it's physics-based algorithm, it's not AI, so it's not blind. Moving on to life without labels. This is where I'm creating new ground, at least in microscopy community, and hopefully this ground will pick up and find its place. So the results I show here will not be the most appealing, but the results will be insightful about where we might be heading, where we have an opportunity window. This is a not so nice looking image, but I've taken it from this paper which says that different components inside cells may be characterized by different refractive indices. And if we are able to image the refractive indices, we have the 3D structure of the objects, and we also have hopefully super resolution if we have sharp boundaries and accurate reconstructions. So the first part that we did here was to model what a coherent microscope is imaging. And it's important and different in the sense that usually in label-free modeling, people use scalar point spread function, especially in incoherent ways. But we have changed it, we have gone full wave, and we have also included multiple scattering. It is quite important, as I will show you through some examples. So what we have done is we have included, first of all, using dyadic Green's function. Dyadic Green's function is like usual point spread function is one number or one value for between sample and a camera pixel, but here we have 9 values for that we take the XYZ orientation here and the XYZ orientation here, and then we take the combination. But also, we have included much more sophisticated substrate geometry. If you have, for example, multiple layers, if you have any other thing that is there, we can incorporate it, and then the multiple scattering solver. And our framework is generalizable enough that you can compute an equivalent bright field image or a dark field or interferometric or other kind of coherent microscope images very simply. This is an experimental result to compare our simulations with experimental data. So this is incoherent bright field image of beads. I think they were 1 micrometer in size. Yes, 1 micrometer polystyrene beads. And we used air immersion, so the refractive index contrast is pretty high, and this is the image we get in incoherent bright field. We do center finding, and then we estimate their locations and sizes. And based on this, we simulate how it would look like in a coherent bright field system or a coherent interferometric system. And for the same region, for the same beads, we have these images and we compare them. So this is important. This coupling that is present here is absent here, and we can replicate this coupling. So this is near field interaction. Also, the relative contrast between these 4 stripes is important, which we also see replicated in our simulation. Why does it matter so much? Really, why? The reason is it holds clue to why our images look like they are. Do you see that this is slightly elliptical versus pretty spherical? This is simulated using full wave scattering, and this is simulated using an approximation called Baum approximation. And if you include full wave scattering, you basically include how your electric field might have interacted in all the 3 dimensions. Now in this case, yeah, it looks like maybe there is something in Y and Z, but how important it is. This is number 4. I'm sorry for not having bigger fonts here. And this is 0.7. This is even smaller. So it seems like it may be negligible, but is it really so? Consider this case. We have 2 beads, and this is the image under a coherent microscope. We can show that, yeah, there is some loss of resolution because of the currents or the interaction along X, but the deal breaker here is Y. The interaction that is inducing fields to be aligned along Y direction. On the other hand, look at slightly more separated beads. We see an artifact in between. This artifact cannot be explained by Baum approximation because it would give 2 flat circles here. But here we see that the reason is Z orientation of the dipoles. This Z orientation is the one that is causing this artifact. And of course, this is a simpler case. We have 2 beads and their images are elliptic, and we see that why they are elliptic. So this is a no-brainer. So clues about artifacts and coherent microscopes are in the near field interactions, which need full multiple scattering solver. We now take on the next challenge. We want to estimate the refractive index contrast. We need to solve for the refractive index contrast. We have put a new microscope in place. As compared to all the microscopes where light comes either from top or from bottom, we have gone for side illumination. We use optical fibers to give side illumination to the sample. This is similar to, for example, you're using inclined beam, right? So it is essentially emulating a dark field here. But the idea with side illumination was that side scattered light is rich in multiple scattering, and this is what we want to use. We created a simulation sample, a phantom sample, and then we computed images under different illumination directions. And then we started adapting algorithms from radar imagery to this domain. So basically what we did was, this is the Baum approximation results, but we took 4 algorithms which are nonlinear inverse scattering ones. They work for radar images where we can have phase measurements and intensity measurements, and there are other flexibilities but other limitations. We optimized it for microscopy domain, and we saw some good results. We saw that CSI gives very sharp boundaries. These are just acronyms of different methods. CSI gives very sharp boundaries, but its reconstruction along Z is poor, so it's not able to use the Z direction information pretty well. There are theoretical reasons for explanations for that. I'm not going into them today. But this is the one that works for weak scatterers. The other side is the one that works for strong scatterers. And although a contrast of 0.6 is very high in the optical domain, in radar imagery, this is sort of medium to strong scattering region, and these methods on top use frequency domain information. So this is what is very valuable. Okay, so we did some computational experiments to test our hypothesis and to find out what adaptations work well. We also did a sensitivity analysis. Are our algorithms robust to not having accurate information about our system? What if sample was lower than where I thought it was, or higher than where I thought it was? In those cases, this method still performs much better than everyone else. So we chose this method, and then we optimized it further for experimental system. These two, and then we started using only this based on an approximation that ours is side illumination, transverse electric case. Now these are our first experimental results, unpublished, but I am happy to share them here. So this is the incoherent image, and it shows that there is one bead, 1 micrometer size bead. These are the images of that bead from different directions. This may actually be looking like 2 beads, but it's not, even this. It's a single bead, but the illumination and the multiple scattering makes it look differently. Born approximation does tell that there is one bead, but its reconstructed contrast is lower, and the reconstructed shape is less accurate than when we use multiple scattering. But when we go for 2 beads, this is where things become interesting. So in 2 beads, we start seeing a lot of artifacts in born approximation. The reason is it neglected multiple scattering among the particles. And on the other hand, CIE can actually do even better job at reconstructing the refractive indices more correctly, simply because it incorporated multiple scattering between them. But of course it fails somewhere. We cannot always just be happy about where the results work. So the place where it fails is when the beads are closer. When the beads are closer, we see this artifact that comes up. The reconstructions are still much better than the Born approximation, no doubt, but this is where we were intrigued, and I will tell you what happens here. This is a sample with many beads, right? We are looking in a small region. This is a 5 micrometer by 5 micrometer region. But what happens is that there are beads in its neighborhood, and we have side illumination. So there is light coming in, there is a bead, and then this bead throws this light forward. This is called forward scattering. Now, the light thrown by any bead forward has a range of 20 micrometer. So basically, we have extra light coming from any bead in 20 micrometer radius from here. These are like headlights of different cars, all pointing in different directions and influencing each other. So basically, taking multiple scattering between 2 closely placed beads is not sufficient. We need to incorporate the cars around in the vicinity. So now we are upgrading our algorithm to take into account extra multiple scattering from far vicinity, or not close vicinity, but a little bit extended vicinity. Hopefully we will show you some very beautiful results later. But with this, I come to my original slide where I introduced computational microscopy. This is where we want to work with. We want to combine these three to make this puzzle. And we are a group, so I think this was our annual day, and you see how seriously we work together. But then there is food, and what people are doing is actually playing a game. So we are a very fun group. We take our play very seriously. Yeah, this is where you can find more about